Understanding your genetic risks for schizophrenia. About 1% of the population is at risk for developing this disorder. However, as we will discuss later, different factors can make this number much larger for certain individuals. Both environmental and genetic factors play a role in causing this disorder. According to recent research, 80% of risk in the general population comes from genetics, while only 20% comes from the environment. This has been proven by studies in which the correlation of schizophrenia between identical twins who have identical genomes is only 48%. Schizophrenia impacts both liver cells and red blood cells. In addition, schizophrenia has been linked to over 100 associated risk loci, and currently there are more than 20,000 possible causal single nucleotide polymorphisms attached to this disease. This means on a SNP chip test, over 20,000 DNA sequences should and could be included. There is not a definite test that can be performed to allow a person to know whether or not he or she is going to develop schizophrenia later in life. However, research currently being performed at the Indiana University School of Medicine has developed tests that accurately predict whether or not a person will develop the condition based on genetic factors. According to the lead researcher, Alexander B. Nicolescu, Associate Professor of Psychiatry and Medical Neuroscience at the IU School of Medicine and Director of the Laboratory of Neurophenomics at the IU Institute of Psychiatric Research. At its core, schizophrenia is a disease of decreased cellular connectivity in the brain, precipitated by environmental stress during brain development among those with genetic vulnerability. A 2013 study states that there are around 22 gene regions that can contribute to schizophrenia and around 8,300 SNPs. Research has failed to narrow the cause of the disorder down to one specific gene. However, C4 genes are commonly associated with being linked to the disease. The complement component 4, or C4 gene, which codes for the C4 proteins C4a and C4b, is the gene that most greatly determines risk for schizophrenia. If this gene produces excess C4a proteins, then the person is at an increased risk of more than 1% for developing schizophrenia later in life. The C4a proteins aid the elimination of synapses and synaptic pruning, which can contribute to schizophrenia and a loss of gray matter. In addition, RS412948 is a SNP located in the pseudoautosomal region near the CSF2RA gene. It has been proven in a whole genome association study, which is the most credible, to be linked to schizophrenia. This risk allele is the G. The odds ratio is 3.23. This study unearthed that rare deleterious variants may be more prevalent in schizophrenia disposition than polymorphisms. A study from 2015, which interviewed young adults at high risk for developing psychoses, voiced the concerns of those deciding whether or not to be tested. Among these concerns was test reliability, data interpretation, stigma, psychological harm, family planning, and privacy. Since genetic testing is likely to only predict relative risk, researchers fear individuals can misinterpret their test results and seek improper or unnecessary help. In addition, results could negatively impact mood and make individuals more susceptible to discrimination. Because schizophrenia is a mental disorder that results in a disturbed thinking, the primary ethical concern regarding schizophrenia is whether or not the potential research participants are fit to make decisions and choose to participate. Participants with schizophrenia are thought to be in a very vulnerable position. It can be hard for them to make informed decisions on participation and medication. In summary, a genotype test could be beneficial for recognizing this condition early on and could help evaluate a predisposition towards schizophrenia.